Hi everyone, so today we're going to take a look at the paper Chemical and Genetic Rescue of an EP300 Knockdown Model for Rubinstein-Taybi Syndrome in Zebrafish by Babu et al. So before we really get into the meat of the paper, I think it's important to go over a couple little background information and some context. So firstly, of course, what is Rubinstein-Taybi Syndrome? It is a rare congenital condition characterized by a multitude of factors, including things like short stature, broad thumb and toe. These are typically also angulated, as seen in Figure C and Figure D. Also intellectual disability, heart and kidney defects, dental and jaw-related issues, and microcephaly, or a small head and brain. And so our current understanding of Rubinstein-Taybi syndrome and how it really develops is not well understood, but we do know a couple of things about certain genes, particularly the CREB and EP300 genes. These are genes that are part of a transcriptional coactivator family, and they play redundant roles within development. So these genes are involved in chromatin remodeling, and really their key role is histone acetyltransferase activity. And this activity is done by this CAT domain over here in blue, which stands for the lysine acetyltransferase domain, in particular in EP300, which is the version that was looked at in these papers and one that we're going to focus on, not really so much on CREB. And we'll get into a little bit later about what it actually does. But for the most part, what we need to know is that these genes are really important in cell proliferation and cell differentiation. And RTS is inherited by an autosomal dominant genetic inheritance pattern. So really one altered copy of the gene is sufficient for the syndrome. What this means is essentially one copy of EP300 will not function or will produce like a reduced amount of protein, which eventually leads to the developmental issues that we've mentioned before. But actually what's interesting about Rubinstein-Taybi syndrome is most cases are actually due to spontaneous mutation and not due to familial history. So they're not really due to the common autosomal dominant genetic inheritance pattern like you see in the figure on the right. It's really just like spontaneous mutations that show up that lead to the disease. And this is most likely mainly because people with RTS just don't have children, and so as a result they can't really pass on their genes. Because our RTS is a genetic condition, there is no real treatment for it. Everything that occurred during development, so there's no real way of actually treating it once the person is born. But there are some other things that can be done with individuals with RTS, for example, physical treatment for the jaw-related and dental-related issues, just to ensure that the person can still, chew and eat properly, is something that's pretty important. And so because we have no real treatment, research really is looking into prevention beforehand, like within the womb, to ensure that EP300 activity, or CREB, for example, is still operating. And that's really where our research comes into play. Modeling RTS in model organisms is actually a lot harder than one would think. So for example, yeast homologs of EP300 share little sequence similarity with human EP300. And so conclusions based on these studies would be iffy at best. And additionally, mouse models, knockouts of EP300 actually lead to unviable embryos. And so we can't actually grow up these mice to look for signs of RTS. There are some conditional knockouts of EP300 in mice that are important for immunological research, but as far as RTS is concerned, we can't really use either of these. So it really means that we need to find some other model for RTS in order to do research. And so based on these issues with modeling RTS, this is really where our researchers come into play. They noted that there's these issues with modeling RTS and all of our favorite model organisms, and so this led them to investigate EP300 homology within a variety of organisms, and eventually they landed on zebrafish, another important model organism in developmental biology. They really were asking the question, okay, could we use this as a model? And so this led into kind of three areas of research that they focused on. One is, of course, the similarity of EP300 between zebrafish and humans. Additionally, they also wanted to see whether zebrafish with EP300 knockouts showed actual similarities to RTS phenotypically. And then additionally, can we use this for potential treatments or preventions and future research? And so hopefully now we can figure out whether zebrafish act as a good model for Rubinstein-Taybi syndrome. And so to start their research, they first looked at the EP300 similarity between humans and zebrafish. It's important to note that zebrafish have two similar but different types of EP300. There's EP300A and EP300B. So specifically, there are a bunch of different domains within EP300, but looking at the lysine acetyltransferase domain is where these researchers focused on because it seems to play the most important role in RTS. So they showed, firstly, through a bunch of database sequencing, that the human and zebrafish EP300A share 89.72% sequence similarity. And actually, interestingly, within zebrafish, it's actually slightly less than between a fish and human, which is kind of interesting. But nonetheless, it just does show that there is significant and important similarities between the cat domains in both humans and zebrafish. And so hopefully we can have some good conclusions based on using zebrafish as a model for human. So next, the researchers actually looked at the similarities in RTS with zebrafish. And so at first, it's important to note this C646 is an inhibitor of EP300. And so what they did is they first showed with the control normal growth of zebrafish, and then they eventually upped the amount of inhibitor in the system. And that showed one, in the red here is the production of the neural tube, and they show that with increased inhibitor of EP300 you have reduced neural tube formation, which is an important aspect of development and RTS. Secondly, they show that there's decreased growth associated with less EP300 activity, again important with like short stature, for example, in RTS. And then lastly, they showed here's a normal jaw growing in zebrafish, and then as you increase inhibitor, you see decreased and almost non-existent jaw formation. Again, important for like the dental and jaw-related issues associated with RTS. And lastly, they also showed a normal heart developing versus issues with the heart, specifically an edema forming as more EP300 inhibitor is added to the system. And really, this does show that there are similar developmental issues in zebrafish as humans with RTS. So alongside the studies using the inhibitors, they also did some knockouts of the EP300A and EP300B in zebrafish. And so again, we see normal development of a jaw as well as heart in regular zebrafish development. However, once we knock out EP300A, we see again this reduced jaw formation and issues with heart formation. Interestingly, EP300B knockouts actually seem to show regular jaw development and heart development. But then again, when you knock out both of them, we have the issues associated with RTS development. But it does seem that EP300A does seem to play a bigger role than EP300B in the development of RTS, which is kind of an interesting note as far as modeling in zebrafish. Additionally, in this other figure here, they show an actual closer fluorescence view of heart development with the ventricle and atrium in a normal case. And you can see when the issues with RTS involve a significant increase in the atrium size compared to the ventricle in both cases where issues with heart development occurred. And so lastly, researchers showed that they were able to actually rescue the RTS phenotype. So firstly, we're looking at a Alcyon blue stain of the jaw structures within zebrafish, and they're staining specifically the molecule SOX9, which is an important marker and molecule in the development of craniofacial structures and skeleton, so in this case the jaw. And as we see with EP300A and B knockouts, we have reduced jaw formation. And so what the researchers actually did is they took the cat domain specifically out of EP300 and injected the RNA for that domain into the fish, in the hopes that it could revert back the issues related with the knockout of EP300A and B. And so in this case it actually did. We see increased SOX9 expression and jaw formation due to an injection of cat RNA. And it actually also showed that it didn't affect normal development, which is an important note. And then the researchers also put in mutant cat RNA and showed that it couldn't actually revert back the RTS phenotype, specifically for the jaw, and it actually reduced growth in the normal case. But ultimately, this does show that there's the potential to rescue RTS phenotypes during development, at least in zebrafish. We don't know about humans yet, but it's an interesting first step that we could eventually use to look at future potential therapeutic and I guess preventative is the better word, things for RTS. And so really to finish things off, it does seem like zebrafish are an excellent model for Rubinstein-Taybi syndrome. Not only does the EP300 in zebrafish share similar sequences with the human version, but the zebrafish actually also show similar developmental issues associated with the syndrome, including the jaw-related issues, heart-related issues, growth, stunting of growth, and neural tube formation, for example. So really it does seem like it's an excellent model. Of course, this does also bring up some, I guess, future research ideas because this is kind of more of a preliminary figuring out whether this is like a good model. So some things I thought of, for example, are specifically looking at molecular mechanisms behind how injecting cat RNA actually rescues the jaw phenotype, for example. And then additionally, this also brings up other questions, such as what's the importance of the other domains within EP300 if only injecting cat domain seems to be enough to rescue this certain phenotype. Obviously, like we showed in EP300, there's a variety of other domains. And so just because we insert cat domain doesn't mean that those other domains have other functions. So what other, I guess, developmental issues would be, I guess, left as a result of not fixing, I guess, the main problem, which is the lack of EP300 activity as a whole. Additionally, they only showed rescue of the jaw. However, it'd be interesting to look at whether you could also fix like the heart issues, for example. And then lastly, it'd also kind of be interesting to note whether there are behavioral changes in these fish. It is kind of hard to, I guess, compare the behavioral differences in fish and humans, but it would still be an important note to make considering we would like to use zebrafish as a model for RTS. But with that, I want to thank you guys for listening and hope you enjoyed.